different words. Thank you. Recorded. Okay, and each word has a different meaning. So sentence parsing can be done very deterministically, but RNA and protein on DNA, you just got A, C, G used. You know, when you heard a word like the, you know you're starting a noun phrase, right? Even though you don't know when that noun phrase would end, but you know you're starting a noun phrase. When you see an A or C or G or U, you have no idea what's going on, whether it should be paired to somebody on the right or somebody on the left, or should be unpaired. You don't know. So you need to do a lot of search on the ambiguity. The search space is much bigger in biology. So that's great. You know, my algorithm can have a new life from language to biology, right? So I did the LinearFold and linear parsing, which I will talk about briefly. But something really unexpected happened. That's COVID in 2020, and that changed everything, right? And I started to focus on COVID since COVID-19 started. And I will talk about two kind of very interesting stories using parsing algorithm, in particular lattice parsing, to help doing mRNA vaccine design. That is, to design better mRNA vaccines than those which we have got like Moderna and Pfizer shots. And it's currently used by a lot of vaccine companies, and it's under revision stage at Nature. And then another story, which I'll probably talk very briefly, is synchronous parsing, which you can use to do joint align and fold. Because synchronous parsing is indeed joint align and fold of different language like Chinese and English, Chinese and Japanese, whatever translation pairs by text. You do synchronous parsing on it. You can do the same thing on SARS-CoV-2 variants like Delta, Omicron. They're like English and Chinese. They're similar languages, or you can think of that way, and you can fold and line them together to find their vulnerable regions of the genome. And you can design drugs and to kill the virus. So this paper was published last year at PNAS, Proceedings of National Academy of Sciences. For any questions, please do stop me during the talk, and I can also pause, to ask if there are any questions. But I would prefer that you guys just be as interactive as possible, as like a real talk, in-person talk as possible. Thank you. Okay, so yeah, so why, first of all, RNA folding is the same thing as natural language parsing? Well, the input is just ACGU sequence, very long sentence. The output is for each nucleotide or each word, you tag it as either left bracket, meaning it's paired with somebody on the right, or a right bracket, which means it pairs with somebody on the left, or a dot, which means it's unpaired. That's just that simple. And it has to be nested, well-balanced, because we assume no crossing pairs. Crossing pairs do exist in nature, but very rarely. It's like, non-projective parsing, non-projective structures do exist, but not too much, right? So let's ignore them for now. So you can see they're just nested hierarchical structures, which is, just a parse tree. Like these dots, these nodes correspond to these pairs, and these unpaired, like leaf nodes, correspond to these unpaired nucleotides for this structure. And say, oh, that's just a parse tree, so we should apply the same algorithm. And indeed, they used exact same algorithm, same grammar to describe and parse these structures. So as I said, you have English sentences corresponding to sequences, and syntactic structure corresponding to secondary structure. Syntactic parsing is just RNA folding. And what algorithm do we do for parsing? Well, CKY, which was like 1960s. And it's a cubic time, very slow, bottom-up dynamic programming algorithm. You know what? Exactly the same algorithm, but using a different name, under a different name, called Nusinov-Zucker algorithm, was reinvented rather, in the RNA field in 1980s. And it was exactly the same algorithm, totally isomorphic. They probably did not know that CKY existed 15 years ago. Like, we were always about 15 years ahead of biology. Like linguistics, computational linguistics, NLP is always quite ahead of biology, and a lot of things can be applied to biology, like transformer, BERT. Nowadays, there's a lot of application of that direction as well. Yeah, but that was too slow. Cubic time bottom-up parsing was way too slow, both for language and for biology, and even more for biology, right? But they got stuck there. They got stuck there for 40 years without knowing that we have made progress in our field. For example, I did, linear time incremental dynamic programming parsing in 2010, which is, very fast linear time, and also explores exponentially many possibilities or alternative structures because it does dynamic programming. And what I did was just apply that, adapt that into the RNA field, and we got LinearFold, which is the first major speed up in RNA folding area for 40 years, because they got stuck there for 40 years without knowing that there's much faster in natural language. That's, I just reconnected the dots. And you can see the results, like for SARS-CoV-2, you can reduce the running time from 1 hour to only half a minute, because we are linear and the other ones are cubic time. And more interesting, they actually, LinearFold, even though it's approximate search, like use beam search on top of dynamic programming, it's actually more accurate than exact search on the longest families, the longest sequences. And that's something we also observe in natural language parsing as well, that even though like linear time parsing using beam search is inexact in terms of search quality, you make search errors definitely, but actually you end up doing better in terms of accuracy by comparing to your tree bank. This is because your model is highly inaccurate. And by doing beam search, you actually kind of doing something like regularization. But I will not get into that. I'll just say that it's very interesting that we're actually doing better, especially on long sequences and long range pairs. Long range pairs, you can imagine they're just like, in a sentence, some words are very far apart, but on a dependency tree, they are actually close to each other. For example, like, what do you serve chicken with? This WH extraction, you should be like saying you serve chicken with what, but what is fronted. And there is a dependency link between the preposition with to the first word what, right? But that link is really, across the whole sentence. These two words are far apart on the sequence, but together or close together in the parse tree, in the syntactic structure. So those things do happen in RNAs as well, that the two ends of an RNA sequence are often together, often very closely together, even though they are very, very far apart. Even for SARS-CoV-2. Remember, SARS-CoV-2 is like 30,000 nucleotides apart, long, that sequence is so long, but their ends do fold together, just like our dependency trees. And our work, LinearFold, is better at predicting those long-range pairs, even though they are much, much harder than short-range pairs. Like local dependencies are much harder, but much easier to predict. Like if you have a determiner and a noun, or an adjective and noun, those are very, very deterministic. But between the main verb and the kind of the object, those are long distance links. They're generally much harder, but we are doing much better on that. And how do we do that? Well, how to fold RNAs in linear time? That's basically exactly the same as how we parse in linear time for natural language sentences. Well, the naive idea is that for each nucleotide, you just say it's either a left bracket or a dot or a right bracket. And left bracket is basically push to the stack. And the dot is just skip that, don't push to stack. And if you have a right bracket, you just basically pop stack, right? So you just maintain a stack, because you're doing context-free grammars, you have to maintain a stack. And that naive algorithm, unfortunately, is 3 to the n. It's exponential, right? It's exponential because every point you just expand to 3 different alternatives, and that's too bad. And we want to do much, much faster, right? What we could do is dynamic programming. And how dynamic programming works is to pack or merge equivalent states, right? So that's the whole point of sharing common subproblems to save computation. Let's say that this state and maybe this state are kind of equivalent under some equivalence relations, which I will not talk about in detail, but let's just assume that there is some equivalence relation under which you can pack these two states, and then you can do some magic that reduces from 3 to the n. So this is 3 to the n exponential. Sorry, wait, what's going on? Oh, are you guys still there?
Yeah.
Oh, okay, sorry, I got disconnected, for example, for some reason. My network got but I'm back. Do you guys see my mouse?
Yes, we're fine.
Okay, sorry. Yeah, I was saying that you can, by packing equivalent states, you can reduce the space from 3 to the n exponential, this is exponential, to n to the 3, or cubic, which is polynomial, right? So now you're back to cubic time as if you're the same as CKY, but this is very different from CKY in the sense that this is left to right. It's not bottom up. So this is kind of incremental parsing, but in cubic time, which is already much better than bottom up cubic time because human parsing is incremental. Just like as you hear me speak right now, you're doing incremental parsing in your brain, even though you don't realize it. But there's no way that you would say, oh, I have to wait until you finish a sentence and I hit a button and say parse. There's no way you can do that, right? Or if you read a newspaper, you scan the sentence from left to right. There's no way that you would say, wait a second, hold on, I haven't finished the sentence yet. Oh, wait until you see the period. Oh, then parse. That's definitely not the case, right? So you parse incrementally, just like RNAs and proteins, they fold incrementally as well. We'll come back to that point. So this is also more like natural folding. Also, like incremental parsing is more psycholinguistically meaningful or plausible, right? So, but on top of that, we want to still be linear time. So what we do is beam search. But this beam search is on top of dynamic programming. So it's very different from the classical beam search like you guys do in encoder decoders nowadays in the beam search. But those beam search is not with dynamic programming in the sense that they only visit constant number of alternatives. Like if your beam size is 10, you only explore 10 or different alternative paths. But with dynamic programming, even with a very small beam size of 2, you can see you already packed exponentially many paths. You already visited those exponentially many alternatives along the way because of merging, packing, and unpacking, right? So the beam search, because of the underlying dynamic programming, is so powerful that in linear time, it can visit exponentially many alternatives because each point here corresponds to millions of different alternatives with the without dynamic programming. That's the essence of the algorithm. So for example, you can pack these two states to this state, and sometimes you unpack if you want. Okay, and let's compare with local folding. Well, local folding has been a popular alternative to global folding because global folding was too slow. Like I said, for 40 years, they got stuck with cubic time folding, but that definitely doesn't work for very long sequences like COVID. So what they did in biology was kind of a quick and dirty solution. They do local folding, that is, they restrict the pairs to be within the local window, like say 100 nucleotides apart. So they can only have, you can imagine that you can only have dependency links between words that are only within 100 words apart. They cannot have very long pairs, very long dependency links, in other words. But that definitely doesn't work, right? Because like sentences, RNAs and proteins tend to have their ends meet, tend to fold into a very compact structure. In other words, the beginning part and the final part of the sequence tend to come together and to have very short distance in the syntactic structure or folded structure. So this is also the case with SARS-CoV-2, I said. And those previous local folding methods definitely cannot catch those. For example, this is a local folding method, and you can see that you can only find very local pairs. Whereas in reality, these in gray are the ground truth pairs. Those very long distance pairs, especially the ones between the beginning and the end, you can never find, right? Our algorithm can find those very non-local, even end-to-end pairs, even though we also make a lot of mistakes in the long-range pairs because long-range pairs are just hard to predict anyways, but it's much better than local folding. So LinearFold is the first linear time and global folding algorithm, so which will enable global analysis on SARS-CoV-2, which I will talk about near the end of this talk. And I briefly mentioned the incremental parsing and incremental folding, and I said, just like you hear me speak right now, you're doing incremental parsing, and the way human beings parse is also very similar to the algorithm I described. That is, you kind of do local ambiguity packing along the way, even though you don't realize it. So there is actually psycholinguistic evidence to support that. It's a mechanism called delayed disambiguation. For example, if I give you two sentences, John and Mary had 2 papers. You say, okay, fine, you just had 2 papers. Until I reveal to you the last word, each. John and Mary had 2 papers each. You were surprised. You were like, wait, I thought they were co-authors. They're actually not collaborating, right? Or if I say together, I say, oh yeah, I know, they're collaborators, right? But before I reveal to you the last word, well, even aware of the fact that John and Mary had two different readings, at least two different readings? Most people will say probably not, right? John and Mary, whatever, just a noun phrase, just two people, whatever. Their relationship, I don't care. For the rest of the sentence, John and Mary just a subject noun phrase, right? So that the local ambiguities within John and Mary is not very important for the rest of the sentence. So that's packing. That's just like, so this is like John and Mary, and basically this state is like John and Mary, and then have two papers. And until you see the word each, you probably have to unpack. So you do packing and unpacking along the way. And that's how you can achieve, you can visit exponentially many alternatives in linear time. That's essentially the whole idea of the algorithm, which is very similar to human psycholinguistic evidence. And that's exactly the same way that RNAs fold too, right? Because when RNA's prefix is being synthesized, they have to fold onto themselves. The chemistry just doesn't allow you to say pause, to say, wait, hold on, you're not done yet, until you have a full sequence. There's just no way to do that. Just like there's no way for you to say, hold on until you finish the sentence to parse. There's just, nature doesn't allow you to do that. So there's very interesting connections between the two paradigms. Any questions before I move on to the next part? Okay, so I talked about parsing. So one best parsing is bottom-up cubic time, and I linearized it. But there's one more interesting connection between linguistics and biology, is that there's ambiguity in our sentences. Like I saw a boy with a telescope, definitely has at least two different readings depending on the PP attachment. Similarly, each RNA or protein sequence has many millions of different structures at equilibrium, meaning like at equilibrium, they fall into a distribution, which is called a Boltzmann ensemble of millions of different structures, exponentially many different structures, and each structure has a kind of a probability in the distribution. So we want to capture the whole ensemble, not just the one best, not just the Viterbi, right? We want the whole ensemble of all possible structures. And that's something we also study in natural language. It's the inside-outside algorithm, which captures the partition function. And this whole thing is widely used, like when we did like conditional random fields and stuff like that, HMM, of course, inside-outside is like more generalization of forward-backward. It's on context-free grammars. But this is cubic time as well, and it's bottom-up and top-down, but replacing the maximization or minimization with a plus, with a summation. So this is kind of the partition function version, and this is the Viterbi version. You know what? The same algorithm has been reinvented again after about 11 years in biology, called McCaskill algorithm. It's essentially reinventing inside and outside. And it's the same cubic time, bottom up and top down. And it's way too slow. It's even slower than the Viterbi version. So I went ahead and did linear partition, which is basically the partition version, the summation version of the LinearFold. LinearFold is the Viterbi version of the kind of the one best parsing version, and this is the ensemble version. But interestingly, there's no correspondence in natural language, because I didn't do that in natural language first. I just did it in biology first. Maybe you can apply it back to natural language, and you can do fast kind of conditional random fields learning for natural language parsing. That's also possible, the other way around. Okay, so I'll probably skip that. It's just, the difference is Viterbi is the minimization of free energy, or CKY, the best one, the one best, and here you sum over all the exponentially many ones. And so in Viterbi, you just do the left to right instead of bottom up, but in partition function, instead of bottom up and top down, what you do is left to right and right to left. But those are not forward and backward, those are actually inside and outside, but we just do inside and outside in the left to right way. And it's much faster than the baselines. And we're also doing more accurate predictions. Just, again, we compare with local folding. Local folding is so local that they can only consider very close pairs, especially on the long sequence. And we can do a lot better. Yeah. Any questions before I talk about COVID? Okay, good. So now the exciting thing happens that, well, unfortunately, COVID hits. So soon after we published these two papers, COVID hits. And in January of 2020, I've been thinking, like, how can my research help fight the pandemic? I know that LinearFold is going to be useful because this is the longest RNA virus. You can enable structural analysis for the first time, but we want to do something more direct. but I'm not a biologist per se. I'm not a medical scientist, so I have no idea how to apply my work more directly to fight the pandemic. So I asked my friend Ryu Das of Stanford Medical School. So he's a biophysicist, and he's been using LinearFold for quite a few years before the pandemic. I said, anything we can do for COVID, except that it was not even called COVID at the time. It was still called 2019 nCoV. And he said, maybe an RNA vaccine. Yeah, he suggested me to look at mRNA vaccine. So he told me how that thing works. I have no idea what mRNA vaccine means. At the time, I have no background in vaccine or immunology. But very luckily, we generalized parsing into lattice parsing, which was a very old idea in NLP. And lattice parsing can solve the exact same problem that we needed for mRNA vaccine design. So we just realized that the connection between two very different problems and used the same algorithm for lattice parsing, which was like 60 years old, to solve the new problem, which is exactly the same problem as the problem in NLP. That connection between language and biology is totally unexpected. So just to give you some basic idea of mRNA vaccines, so everybody has been injected these Pfizer and Moderna vaccines. But what's the story behind it? Well, the story behind it is very simple. So instead of the protein vaccine, which is injecting the executables, the proteins, meaning the executable files into your body, we inject source codes. Okay, mRNA is the messenger RNA, is the source code, which can be translated or compiled in your cells, in your body, to become the executables. So in other words, we give you the prescription, but not the drugs, and let your body be the drug maker for your own drugs. Why is that good? Because you don't need a real virus, to produce the vaccine. So in fact, Moderna started working on mRNA on COVID vaccine in January of 2020. That was before the first case was reported in the United States, right? That was long before that. But because the source, the sequence has been already available online, Moderna or other vaccine company just need to extract a certain piece of the source code and say, oh, the spike protein source code, oh yeah, just make that a vaccine. So it's much faster. You don't need to deal with the virus. Deal with the virus is very, very dangerous. You don't need to deal with any virus or weakened virus or dead virus. It's very fast for mass production. That's why more mRNA vaccines were made available first during this pandemic. And there was no risk of infection. Whereas the protein vaccine, like flu shots and others, traditional vaccines, they're all protein-based. They have a small risk of infection because they're basically weakened or dead form of the virus. But what if it's not weakened enough? It's not sufficiently weakened, it's still kind of active, and then there's always a small risk of infection. But there was always a catch. The catch is that mRNA vaccine is largely unstable. And that's the major reason also why it's not approved before the pandemic. It has been studied for a long, long time, but it's only during the pandemic that we have like emergency use permissions for those vaccines. But compared to protein vaccines, proteins are much more stable, right, in terms of structure. mRNAs are much more unstable because they are largely single-stranded. RNAs are largely single-stranded. DNAs are double-stranded. DNAs are more stable than RNAs. So RNAs can easily degrade. They got cut down to pieces in your body or before injection or after injection or in your cell or whatever. And after they got cut down to pieces, they cannot translate to the full-length spike proteins. And as a result, you got less antibodies, right? So this is the story, like from mRNA, you translate to spike proteins, and spike protein induced immune responses. But once you have no longer full length, you have less full length mRNAs, you got less spike protein and less lower immunogenicity. So our goal is to design more stable and efficient mRNA vaccine sequences so that you can have higher antibody response. And the problem of mRNA design is actually very simple, a mathematical problem. Because if you look at the protein translation direction, that's from left to right, that's deterministic. You can look up the genetic code. So there, just each 3 letters, every consecutive 3 letters like A, U, G, U, U translate to one amino acid, like AUG translates to methionine, and UU is phenylalanine, and so on and so forth. That's deterministic translation. But mRNA design is the reverse direction. That is, from each amino acid, there are different ways to express that amino acid, right? Because you have 64 triplet codons and only 20 amino acids in the genetic code. So the inverse direction is non-deterministic, right? For example, valine has 4 different choices of the triplet codons. leucine has 6, and this thing has 4, and stop codon has 3, right? And that's a combinatorial explosion problem. You multiply the choices together, and you got a humongous number, right? It's so big. Then this number is so big, 10 to the power of whatever, 600 something, that if you enumerate them one by one, even if you can do it 1 second a sequence, that would take like 10 to the 600 billion years. So this is much more than billion years, right? And these sequences, even though there are so many, they're all paraphrases, meaning they're all synonymous. They all translate to the same spike protein. And every single one is a good, valid vaccine candidate. The question is, which one is the best for a vaccine? Well, it turns out that Moderna published a paper right before the pandemic, so December of 2019 in PNAS, a very important paper that showed convincingly the more stable an mRNA is, the higher protein you can produce. So which is very intuitive, like as I explained on the previous slide, but this was largely unknown or kind of controversial before the publication of Moderna's paper. So Moderna has been working on mRNA vaccines for many years. They're always one of the front runners. But this paper gave us the direction that we want to find the most stable sequence among these exponentially many alternatives. But the question is, how do you find them? You cannot enumerate them one by one. So Riju Das, the Stanford scientist who told me this problem, said, "Oh, I believe it's NP-hard. There's no way you can do it computationally." So his idea is to use LinearFold plus his cross-sourcing game and let human beings design the game, design more stable sequences. He said maybe humans have better kind of intuition of better search. But I said, no, that's not NP-hard. And I realized that NLP can solve it. And the idea is to use word lattice, 2 ideas from NLP, word lattice and lattice parsing. Of course, I didn't come up with this complete solution in his office, but when he told me this story, but after a few weeks, I found this solution very nice, very clean solution from NLP. So the first idea is to borrow the thing called word lattice from NLP. So word lattice is very simple. So let's say you hear a speech signal, but you cannot hear it very clearly, maybe because of noise or accent. So you have some alternatives of ambiguities, like maybe it's I like this veal, or maybe it's I like this meal, or maybe I like this or his veal or whatever. But it's often like similar sounding like homophones, like you like bear and all those things that you cannot fully disambiguate just by listening to this acoustic signal alone. You need a language model, you need maybe syntactic grammars to disambiguate. But anyways, we kind of pack the ambiguities in lattice. We use the same idea for mRNA. So for example, you have methionine, you have 1 choice. So you just a single chain, 2 choices. Oh yeah, there's an ambiguity in the last letter. 4 choices here, valine, 2 choices, and 6 choices for leucine. That's the most complicated one. You can concatenate these little DFAs or lattices together. So this is basically finite state automaton or DFA. And until you have everything, then you have a huge, very long and thin DFA. That's called an mRNA DFA or mRNA lattice, which encodes compactly the 10 to the power of 600 something exponentially many path, right? So that's the first contribution that we find a very good representation for the design space using DFAs or lattices from NLP. But that doesn't solve the question, right? The real question is, how do you find the most stable path within this lattice, right? So this lattice encodes so many different paths. You cannot enumerate them one by one. So how do you find them? Well, that's the second idea, the most important idea in this talk. That's borrow lattice parsing again from NLP. So in lattice parsing, we have a lattice. So we encode a grammar. We have a grammar that encodes grammaticality or likelihood or whatever. And we use some magical algorithm called lattice parsing, which probably people in this audience are not familiar with. But in the old days in NLP, lattice parsing is used quite often in to help speech recognition. It can magically parse all sentences in this lattice simultaneously without enumeration using dynamic programming. After lattice parsing, you find the best path, best sequence of sentence, along with its optimal syntactic structure. You actually parse all these sentences together, but you just can find the best sequence with the best structure. Same idea for lattice for mRNA. After you got me a spike protein, I converted it to the mRNA lattice, which I showed, and then used the exact same algorithm, except that the grammar is different. Now the grammar is the RNA folding grammar. So instead of grammaticality, it encodes stability or free energy. So the lowest free energy sequence that you can find by lattice parsing, which folds all the mRNA sequences simultaneously without enumeration, after which you can find the optimal mRNA sequence and its secondary structure. And so this is like magic, right? You can solve such a hard problem with something very old in NLP. Well, it turns out that lattice parsing is a very simple generalization of CKY, or the single sentence parsing from a single sentence to a lattice. Well, you can imagine CKY being I assume everybody knows CKY. CKY, you can imagine it as a special case of lattice parsing where you have a single chain lattice. Your sentence is just a single chain lattice, right? Lattice is just a generalization from a single sentence to some alternatives, some ambiguities, right? So you can imagine if you only have a sentence like I like this meal, you can do CKY. That's a single chain. That's a special case of lattice parsing. And lattice parsing was actually solved in 1961 using something called Hillel construction by this pioneer in our field, Yehoshua Hillel, who was a pioneer in formal linguistics and machine translation. Nowadays, nobody knows who's Hillel, but actually he's very important in linguistics, computational linguistics, and theoretical computer science. He solved this problem of mRNA design 60 years ago, 60 years before the pandemic. Of course, without knowing the pandemic, but that's exactly the same algorithm we're gonna use. So basically, we generalize from CKY, ij, k to lattice parsing. Well, we just generalized each index I, j, and k to states in the DFA or automaton or lattice to P, Q, and R. So whenever you have B and C, and you can combine into an A with a production A goes to B, C, you can do the same thing so that you can recognize P to Q, P to R become an A if you can combine B and C. All right, so it's exactly the same algorithm. You can also view it as you construct a new context-free grammar G prime from the context-free grammar and the DFA. So it's basically, it's doing intersection on the context-free language and the regular language. So basically it's CFG and DFA together. So that's another interpretation in theoretical computer science or formal language theory, that context-free grammars are not close under intersection, but context-free grammars and regular grammars, you can intersect them and produce a new, bigger context-free grammar. That's essentially Hillel construction. Any questions? Okay, yeah, his algorithm was so useful 60 years later. So the results on spike protein, so with about 10 minutes of parsing, lattice parsing. So this is the wild type. Wild type meaning the virus's original sequence in the virus genome. It's very, very unstable. You can see that it has a lot of single-stranded. The red regions are single-stranded segments, which can be easily cut down into pieces. So it's about 60% paired and very high energy. After about 10 minutes of optimization lattice parsing, you got a completely different sequence with a completely different structure, which is mostly like DNA than an RNA because it's mostly double-stranded. You can see that these parts are double-stranded with very little segments of single-strandedness, right? So it's mostly like more than 80% paired. We can prove that it's the most stable, or in other words, lowest free energy sequence. Even though like, they look very different, they actually mean the same thing. They both translate to the same spike protein. They are synonymous, but they have very different sequence and structure. So that's the blessing of genetic, the redundancy in the genetic code, that you can have so much freedom of design. So you can explore so many different alternatives to design the best sequence. And we went on to team up with a company, a Seminar in Shanghai, which is a vaccine company, and they did wet lab experiments for us. So in the end, they actually did it even for preclinical study, that is animal studies. So they have mice and they inject our designed vaccines. So we designed the vaccine sequences and then they inject them into the mice. 2 shots, just like what we did for Moderna and Pfizer, 2 weeks apart. And after another 2 weeks, we test the blood samples, we collect blood samples and test antibody response. And you know what? We got up to 23-fold increase in antibody response compared to conventional design technologies like codon optimization designs, which is used by Moderna and Pfizer. So this is not compared to wild type. Wild type is even much worse. It's compared to already optimized designs. And I can talk a little bit about the baseline of codon optimization. That is, there's actually another dimension that we need to consider in this problem, mRNA design problem, we only talk about the x-axis, the first dimension, that is the stability or kind of energy, right? You want lower energy, the better, because lower free energy means better, more stable in physics. You want to push to the left. But you also have a y-axis you want to consider. That's the efficiency of translation. That is, for different codons, like leucine has 6 different codons, and serine has 6 codons, arginine has 6 codons, blah, blah, blah. It's not equal, right? Leucine actually prefer CTG and not TTA, for example, right? So every amino acid has a distribution over the 4 or 6 or 3 different codons. So that's called human codon usage table. You can have a different codon usage table for different animal. We want to use the frequent codons that human genome use. We want to avoid the rare codons that human body doesn't like, right? So that increased the efficiency of translation. So that's a 2-dimensional optimization problem, and you use a lambda parameter to kind of balance between the two. When lambda is 0, you only consider kind of free energy or stability. That's the most stable sequence that we just showed. This is the wild type, very bad in both energy and translation efficiency. But if you increase the lambda, you gradually go up, right? And when lambda is infinity, you have a CI of 1. CI is the codon optimality index. And this lambda is infinity, meaning you don't care about stability, you only care about codon optimality. And that's a very easy greedy optimization problem. Greedy algorithm can solve it. So for each amino acid, you just always choose the best or most frequent codon, and then you don't need to do any dynamic programming. You can just achieve that point. But that's pretty bad in energy. And this curve is the feasibility limit, right? You want to push towards the top left, right? Because the left better, the top the better, the up the better. But this is the infeasible region beyond this boundary. And lower right of this boundary or feasibility limit, everything is possible. You have a huge number of candidates that are possible. So this is the feasible region. And previous work, like Moderna and BioNTech, Pfizer, they only look at codon optimization direction, that is from the wild type towards the codon optimality point, right? They just optimize codon. They have no way to go left of this line, because going left of this line would take billions of years if you don't have a correct dynamic programming algorithm. So this whole huge area here is totally uncharted territory. And we tested these samples, and these designs are shown to be a lot better in terms of antibody response than the codon optimized baselines. That's the story. So without too much details, let's just summarize this part. So this part shows that NLP can save lives. So it's a parsing-based vaccine design with lattice parsing to solve the mRNA design problem. It's currently used by a lot of companies over the world, including Sanofi, which is the French giant Sanofi, which is the 5th largest biopharma in the world, who have licensed it with milestone fees when linear design sequences enter clinical trials. Like clinical trial phase 1, they give us a large amount of money, and phase 2, and another large amount of money, and so on and so forth. Pfizer and Moderna are also very interested. But more important, beyond vaccines, we can do mRNA medicine, general mRNA medicine for any therapeutic proteins, meaning that we don't need to just design vaccines. We can design mRNAs for any other drugs. Okay, so this is very important. So mRNA medicine is the future of medicine, in the sense that I always give you the prescription and let your body be the manufacturer of your own drug. So that's totally personalized medicine. So that's going to be the future. So there's CAR T therapy, there's monoclonal antibodies, and there are a lot of things happening. And we received positive reviews from Nature, and it's under revision now. Okay, so lastly, so we talked about a lot of things, lattice parsing. And lastly, we want to talk about very, very briefly is synchronous parsing. So synchronous parsing is used very often in the old days in machine translation and in semantic parsing, right? So in machine translation, you have Chinese and English. They're kind of corresponding to each other, called a bitext, parallel text, and you want to parse and align them together. So instead of just parsing Chinese and parsing English and then alignment, you want to parse and align jointly. So that's the exact same thing that we want to do to parse or to fold and align Delta and Omicron and other variants of SARS-CoV-2 together, so that we can better look at their structures. So if you just look at one sequence alone, like the SARS-CoV-2 reference sequence from Wuhan, you don't see that much information. But if you look at Delta and Omicron and all these variants together, you're able to see more important details like, oh, if this region is conserved, that is, it cannot change, it has not changed in the evolution, then maybe it's a more critical region that you can design drugs for. But if this region is changed so much, then you probably cannot design a drug for it because future variants can quickly evade from that drug. Okay, so that is the paper called Linear TurboFold, which we published at Proceedings of National Academy of Sciences last year. It's the first album that can scale to SARS-CoV, the first homologous folding album that can scale to SARS-CoV-2. So yeah, so our goal is to do linear time, global, and homologous folding. Homologous folding just means fold and align different variants together. And previous work cannot do that because even single sequence folding is too slow, right? What they do is local folding, just like as I said, they consider local windows, which is really bad. And then they can also only do single sequence folding. They cannot do homologous folding. They cannot look at variants together. So our work can solve both problems, but this problem is extremely hard to solve. So let me give you a brief overview of homologous folding. So there are different kinds of homologous folding. You can do fold and align. That is, you fold each individual sequences, and then you have structures, and then you align the structures together. That's like you parse Chinese, parse English, it's independent parsing, and then you align the trees together. Alternatively, you can align the sequences together and then fold or parse the aligned sequences. Okay, that's align and fold. So these are two complementary approaches, but more interesting, you should do the joint align and fold, okay, just like synchronous parsing. But that's extremely slow because that's kind of doing CKY together on two different languages, like CKY square, and CKY is cubic. That's why CKY square is n cube n to the 6 for synchronous parsing. That's why synchronous parsing is very slow, both in language and biology. So basically, it's like the whole n to the 6 instead of n to the cube. So so there's a previous work where we kind of linearize, that is TurboFold, that's iteratively fold and align. So instead of fold and align together, you iteratively fold and align, fold and align, fold and align. So you're folding into soft structures, not hard structures, and you align also into soft alignment. So everything is like partition function, probabilities, marginal probabilities, and so on and so forth. What we did was just linearize the whole process, so we have linear TurboFold. So I'll not go into any details of that. We'll just show you some examples. So if you fold sequences, single sequence folding, then you don't have that much information from its relatives, from its homologs. You got very kind of different structures, very bad structures, very different from the ground truth. This for tRNA. And if you use linear TurboFold to fold them all together, knowing that they're relatives, they're kind of close relatives, you can fold them into much nicer structures. Because the homology actually, the alignment actually can help you parse. And for these two sequences, if you do single sequence folding, you fold into very different structures. But if you do homologous folding using linear TurboFold, you can have much better alignment and folding. So it turns out that you have an inserted branch. So this is like a whole phrase is mentioned on the Chinese side, but not translated into the English side. That's quite common in our bytext. It's not like our bytext or parallel text is always so ideal, so perfect. It's often like in translation, you always mistranslate or added something or whatever. So it's often the case that you have a whole phrase being inserted. So like in the alignment, you have this not very diagonal, you have just a flat or kind of vertical region. So that's the inserted phrase, and you can very nicely do it. Initially, you cannot do it. You kind of are very vague about what's going on, and gradually, after a few iterations of align and fold, fold and line, you are more clear about where the insertion should be. And after 3 iterations, you're almost like ground truth, right? So that shows the example. And what's more important about this is it enables global structural analysis on SARS-CoV-2 for the very first time. So this is the first joint fold and align algorithm to scale to COVID. It's linear time, global, homologous. And by doing that, we successfully detected end-to-end pairs, that is between the beginning and the end parts, so end-to-end pairs. So this is actually about 30,000 nucleotides apart. Their 2 ends actually fold together. I said many times during the talk that even though the sequence is very long, they're so compactly folded and their 2 ends actually meet together. And you know what? The very striking thing is that this region of interaction between the beginning and the end-to-end pairs, match exactly, pair by pair, exactly with a purely experimental study published by Ziv et al. 2020. And all previous efforts can only do local folding, and they cannot predict this long range of global end-to-end structure. They can only do like, oh, within 1,000 nucleotides or whatever. But this SARS-CoV-2 is way too long to use local folding, right? And finally, we want to find the Achilles heels or the vulnerable regions of the virus genome. So we want to find the conserved and accessible regions. By conserved, meaning those regions should not change during the evolution of the virus. Like whenever new variant comes, this region should be conserved. And also they want to be accessible, but I will not explain that. They want to be, like they can be attached by the drugs. So they want to be the accessible regions, but I have no time to explain the details. I just show you the results. So also we need the regions to be long enough so that it's more specific. Otherwise you might target your own human gene, right? So we want it to be accessible in SARS-CoV-2 and also conserved in SARS-CoV-2 and also conserved in SARS-related. SARS-related are something like distantly related to SARS-CoV-2, such as SARS-CoV-1, those original SARS in 2003, I think. In China, there was like many years ago, there was a SARS-CoV-1 virus, kind of endemic. And there was also bad coronavirus and other coronavirus from other animals. We wanted to be conserved in those SARS-related genomes as well, because they are kind of the evolutionary history of this virus. If some region is conserved, even on SARS-CoV-1, it means that historically this region is so important that you cannot afford to change it. So that it's more likely to be conserved in the future as well. And in the end, we have found 33 regions of interest, which can be used for drug design. And more importantly, our work is published before Omicron. And our data set, of course, does not have Omicron. But you know what? Because we require the SARS-related conservation, that is, our regions have to be conserved historically. It turns out that our prediction is not affected by Omicron. So our 33 regions of interest for potential drug targets are not affected by Omicron variant. But if we loosen this constraint, if we delete that constraint, then the regions that we found will be affected by Omicron. So this means that our predicted vulnerable regions for drug design will likely remain insensitive even to future mutations. Like, for example, Omicron was known after we published our paper, right? So if there's new variant that comes in tomorrow, we'll check it. It's very likely that still not going to affect our predictions, because we look at historically related, kind of quite distantly related, not very close, but quite distantly related genomes. For example, SARS-CoV-1 is only about 80% similar to SARS-CoV-2. So within about 20 years, the virus has changed so much, right? Yeah. And within the SARS-CoV-2, it's only 2 years, and you have not got enough time to change that much. So within SARS-CoV-2, the current mutations is still within 1%. So meaning like even the more distant ones like Omicron compared to Wuhan, it's still within 1% difference. This is a very, very small difference. Okay, so that's the end of the analogy. You also have lattice parsing and synchronous parsing for mRNA design and homologous folding that I talked about. And with that, I would like to thank everybody, with my co-authors and co-workers, and we have the servers and papers. Thank you, everybody. I can take questions.
Thank you so much. Yeah, so maybe if anyone has any questions, yeah, Jia, just go ahead. Yeah, Jia, hi.
Hi, Liang.
Hi, long time no see.
Hi. It's great to attend your talk, and it's really mind-opening to see those connections of genome and parsing, and the results are really amazing.
Thank you.
I have some questions. Maybe they are more basic. The first one is the approximate search with a LinearFold. When it's compared with the N-cube algorithm, would it lose any accuracy?
Yeah, actually, it's slightly better in accuracy, interestingly. You would think it's worse, but actually it's better, slightly better, especially on long families, especially on long sequences. Yeah.
Can you please give some give some intuition or more details?
Yeah, right. Yeah, this is always intriguing, right? So we found that same thing in incremental parsing as well, like doing, beam search with dynamic programming can actually be better than exact search because the model is not accurate. If the model is perfect, that means if you give the best search, you're always going to be 100% accurate, then or the most accurate you can be, then of course you want to be as accurate as possible in terms of search. But because the model, our machine learning model is so inaccurate, there's a lot of kind of noise in the model. So it's not necessarily the case that the best search will be the best, most accurate according to the ground truth, right?
I see.
There's the model accuracy and search accuracy going on. So our beam, sorry, go ahead.
Oh, sorry. So do you mean the beam search actually can have also the function of filtering some noise in the learning?
Kind of the regularization effect, I would say. It's like, it's funny, but yeah.
It's very nice. So another question is about the homologens of folding. So what if we do not consider parsing, but only consider this pure translation problem and learn this sequence to sequence as a sequence to sequence translation without considering parsing? Then would that be less robust, less accurate?
Oh, you want to do folding as translation, right?
Folding as
translation. Right. Yeah, there actually work of applying sequence to sequence models. You can have this as one language and the output is another language. You can think of that as machine translation. But the problem with this is that the output is so structured that you have to have a stack. But of course, we can do that. Even parsing, we can do sequence to sequence parsing. Is quite popular kind of alternative to CKY or kind of real parsing. But so far we haven't seen a very good result in that. Even myself, I probably tried that a little bit. I think that's just a translation problem, right? Yeah. So people are still working on that direction. You can view it folding as translation instead of parsing. Yes. Yeah, you can also do that. You can also do it as a shift reduced parsing, just like we did for natural language parsing as well. There are different ways of kind of formulating this problem as similar problems in our field. Yeah, but that's a very good point.
Okay. So probably the parsing can capture better structure, right?
Yeah, parsing, it's probably better in that sense. Like you can have a grammar, but translation you can have deep learning to help you as well. But of course you can do parsing with deep learning as well. There are papers on doing that as for RNA folding as well. Yeah, it's just that I didn't talk about that. Yeah.
Thank you so much.
Thank you.
Great talk. Thanks.
Thank you. Hi, Kathy.
Hi, Liang. That was a great talk. It's like feels kind of outside of, what I am capable of understanding, but it was really interesting. I'm wondering about the impact. So, you said so far it had been used for the Sanofi vaccine, and somehow it seems like it should be of interest to everyone.
Yes.
Do you see that happening? Or
That's a very good point. Within about 5 years, I think everybody will switch to using our algorithms. Right now, it's just slowly kind of propagating into more and more vaccine companies. Like Moderna is going to talk to me as well. Well, actually, they talked to me a few times. They just say, "Give us the code, give us the code." It was the code, they're very adamant. Give us the code. But the way why the Pfizer and Moderna vaccine worked before our algorithm or without our algorithm is actually another thing called chemical modification, which probably some of you have heard in the news that there was a scientist, Carrico, who invented, also from Japan, who invented chemical modification. So that the vaccine you got injected is not the natural RNAs that we talk about. It's the chemically modified RNAs. And by chemical modification, they can become more stable. So in a sense, it's orthogonal to our combinatorial design. It's like using chemistry instead of computer science to achieve the same goal of stability. But we can combine them in the future to probably have the best results. But right now, the Moderna and Pfizer and everybody else's vaccine use chemical modification, not our algorithm yet. Yeah, that's the current stage. But probably in the future they will switch. Yeah.
Well, really interesting, and you made it very accessible, which was great. So thanks.
Thank you. Hopefully the connection between linguistics and biology is clear, make it easier to understand. Yeah, thank you.
Okay, well, it seems like